The first speaker is Aleksandar Vojta from GlycanAge.
So, good morning. Thank you so much for having me here. I got soaked in this wonderful Copenhagen weather, which is fortunate because I'm going to deliver a talk about a hot topic, which is the glycan clock for biological age. So, the first salient question you might have is, okay, seriously, another aging clock? Why should I care about it? And for good reasons, in fact. So, what we're measuring is a proxy for low-grade inflammation, which turns out to be a very good snapshot of your general health, as we will see a little bit later. And it's connected with the immune system, which goes everywhere. It really comes to every part of your body, so there are no concerns about having the relevant tissue measured. And we analyze the actual mediators of this inflammatory process. So it's not just another biomarker, it is the molecule that mediates an interaction. And we call it the Goldilocks test because it's just about right, not too short, not too long. You get a picture of about 3 months of your health and lifestyle. And the main thing, it is actionable. And for the past decade, we've been hard at work at figuring out which interventions to recommend and how this biological age can be influenced. So let me tell you a little bit about glycans. Now, if you look at these two birds, one is without feathers. And if you try to study something like that, you will find out that you will miss out on a very important property, which is flight. Similarly, this thing rotating in the middle, it's a protein, and all this stuff sticking out from it are glycans. This is the receptor from COVID, essentially. So, as you can see, many interactions are mediated not only by the protein part of the molecule, but by the glycan part as well. So the take-home message would be, if you want to see the complete picture, include glycans in your studies. Now, the last picture is a little bit, like over the top. Here we have the glycocalyx. It's massive, but it shows how much this glycan part can grow in comparison to the plasma membrane and other parts of the cell. So, interactions are mediated both by the glycans and the proteins. Most proteins are glycosylated, meaning anything that is topologically on the outside of the cell. So the endomembrane system, the cell membrane, and anything that is secreted like IgG. And the glycan clock for biological age analyzes the carbohydrates attached to an otherwise protein molecule. It has an obligatory glycosylation site. Depending on those glycans, they are more or less inflammatory. The interaction is mediated by binding to the Fc receptor. So what we're measuring, so the point is here, we're actually analyzing the site where this interaction happens. And how we do it? Yeah, first we bleed you 4 drops of blood, and then it goes into the analysis pipeline. We are analyzing, currently it's 27 different glycan structures, and then there is a statistical model in between your results and the reference cohort. It comes from the Twins UK study from Professor Tim Spector from the King's College London. And this statistical model will give you actually your biological age, more on it very soon. So this is just to show you that when we go back and say, yeah, we analyze glycans, it's very technically involved. However, we do happen to run a high throughput facility in Zagreb, Croatia. It's one of the rare high-throughput glycomics labs in the world. And this is how it gets done. It ends with UPLC, and then we get those peaks, and that's the output. That's the glycosylation pattern of your antibodies. Now, IgG glycosylation correlates with aging. And we might have opted for something like, okay, this is a test for how pro-inflammatory your immune system looks. So why not express it in some arbitrary units and just, put it into a context, into some reference interval. However, it is much more intuitive since it really correlates with age to give you a number that is intuitively understandable. And there is a clear-cut biological interpretation of that number. And that would be if you're older than your chronological age, there is excess of low-grade inflammation. If you're younger, you're good. Let's see what we can do to maintain it in the long run. And what I do is not glycomics mainly, but epigenetics of protein glycosylation. And I just wanted to point out that those epigenetics clocks are based on information. We've heard some amazing talks, and what really got me interested was to hear that information gets mentioned, because epigenetics, it means, you're aging, and the epigenetic information goes out from your epigenome. The glycan clock is based on the effector molecule. So it's, again, and I want to ram this point home, it's like we're not analyzing a biomarker that happens to be associated with it. We're at the interaction site, and we're looking at how it's modified and how this interaction is modulated. And as you can see in this picture, so the immune system goes beyond IgG. There is glycosylation happening all over the place, and as far as we know, it turns out to be quite physiologically significant. Now, again, these functional effectors are associated with different diseases, and depending on how they look, we even have some idea on specific structures and what they might tell you in addition to your biological age. So why inflammation? So if you look at age-related diseases, you can get you have many facets of aging and disease, and the central theme in everything is inflammation. So that is why inflammation tells you a lot about your biological age or health status at the moment. And this is by no means an extensive, complete list. This is just to showcase some of the things that we were able to ascertain, okay, this really shows up in the glycan test. So number one is, of course, natural aging, because many diseases actually resemble natural aging. So I will just showcase a couple of highlights because of time constraints. But we did a lot of these studies in the past decade. And for instance, COVID-19, it's a hot topic nowadays. It's still something we care about. And yes, it does make your glycome appear older, especially if you've been down with COVID and had significant symptoms. So it can help stratify patients with certain autoimmune diseases. Autoimmunity, of course, it will trigger the test. It will show you your biological age through the roof, and then you have an idea, okay, there might be something going on. Then we have like obstructive pulmonary disease, cardiovascular risk, and hypertension. That is quite relevant for aging, we might have some extra info on it. So the first highlight would be the menopause. Now, estrogen is intimately connected with protein glycosylation. I'll show you a bit later how exactly we think it happens. And it is a very good indicator of perimenopause and menopause. And the very interesting thing is we're in an intervention study with hormone replacement therapy. So as soon as estrogen gets replaced, the biological age goes down, and we see it so consistently, we could consider it a highlight, right? Like really one of the more successful things that came from this test. The other is type 2 diabetes. Now, glycans are highly predictive of type 2 diabetes, and we had this study with twins. Now, they're a very high quality cohort with all analysis done to them. Many of them have glycans, so we didn't actually need to run a prospective study. We could go back in time and look at the cohort and figure out whether glycans can predict type 2 diabetes. And it turns out, yes, they can. Which is very significant for healthy aging. And another highlight, cardiovascular health. Now, this particular glycan structure that you see here, it doesn't make it into the formula. The model gives it a weight of 0. However, it might be important for cardiovascular outcomes in its own right. And we're currently designing another product. We're validating it. It's the glycocardio, which will tell you a little bit more about your cardiovascular risk. And of course, there is the gut microbiome, a very important thing. So there are so many interventions out there, and the question is, okay, sounds fantastic. Does it really work? And it seems that the microbiome is very important, because just think about it. So this is the part where it sounds good. It sits at the interface between us and the outside world, and about 70% of the cells from the immune system are there, and they do long-range interactions. Now, these interactions modulate our health in a very significant way. So you might have a sleep specialist who recommends microbiome optimization, because of these long-range interactions and signaling. Now, we're running a study. It's very promising at the moment. So yes, I think I'll be able to say in a very short time, definitely microbiome is a thing. As for interventions, I can, smugly say it's exactly 39% that is heritable. That's the benefit, the boon of the twin studies, because you can really identify the genetic component. And the rest is natural aging and lifestyle, meaning you can do interventions, you have so much wiggle room to do something about it. Let's say your biological age comes back as really, really high. No need to be desperate. You can really rewind the glycan clock. And the whole idea, and what I find is a trend nowadays, of course, that's why we are here. So to catch aging and disease at this point where it's still actionable, you don't feel any symptoms, you're not sick, you're not seeing a GP to help you with something, you just want to stay healthy. And this is where this type of a clock for biological age comes in. Now, it will get triggered while you are at this phase, not developing systems yet, before you hit the point of no return. So what can you do about it? So it's pretty boring. If you do have some health issues, it makes sense to address them either by pharmacotherapy or other means. But otherwise, apart from diseases, smoking is really bad for you. Stress is not good for you. I was kind of skeptical about it, but then a study came back showing stress is really, really not good for you. Diet and exercise. Exercise is kind of interesting. We see a massive improvement upon interventions that include exercise. But then again, we've had some people tested who might represent the 1% of the fittest people on Earth, and yet they are biologically old. So overtraining is a thing. Too much of a good thing is actually pro-inflammatory. It just shifts there. And good sleep is very important, but I'll let you know a little secret that I've been using these days. If you have energy drinks regularly, you don't need to sleep at all. A lot of coffee as well. So, epigenetic regulation. That's what I wanted to talk about. This is like my scientific interest, main focus. And back in 2013, we did something for like a review, and we called this epigenetic regulation quantum mechanics of biology. Why quantum mechanics? It references the paradigm shift that was introduced into physics in the early 20th century, and we wanted to, make it sound good and show how important it is. It might warrant a paradigm shift. So, how is it regulated? Well, it's a network. It's a complete mess, and when you start to tackle it in the lab, it can get frustrating at times. But this is a quite important finding. So this came from GWAS study where we identified some pathways. Now, those orange colored squares are the glycosyl transferases, the enzymes that really directly modify the carbohydrates while the proteins are being synthesized. Of course they're important, but what we wanted to see was how are they regulated? And this regulatory network, which you can see is quite involved and complex, just sprang into the picture, and you can see how it crosses other important pathways. And there you have it, the connection with autoimmune diseases, many of them. So it's there is even Parkinson's, which is an inflammatory disease of the brain, for instance, and this gave us, we started to unravel this network. And I say unravel because it's really like pulling from one side and just climbing along this regulatory network and trying to identify the key factors, the key regulatory factors that make the glycosylation the way it is. This is just to show you how the biochemical part is complex. So while doing research, you can get frustrated because you downregulate one gene and you get a completely different outcome. But when you look at this whole picture, then it kind of starts making sense that you see the effect down the pipeline. And yes, because of their epigenetic regulation, glycans, in fact, integrate environmental information. And what we did relatively recently was trying to remember that network. We found the upstream regulators. RUNX3 might ring a bell, very common, usual suspect. So we tried modulating its transcription, and sure enough, we got an effect on glycans. which is to show, okay, we're climbing up this regulatory network, we're analyzing it, we're figuring it out, there's still a lot of work to be done. And for that, you need to have the right tools. I'm the cloning guy, and back in 2016, we were the first lab to develop CRISPR-based epigenetic modulator. Now we have a whole library of them. We synthesize it, and that's how we prod the genes, like up or down regulate them without knocking them out. So this is like dead Cas9. It just binds DNA, and you have the effector, which does stuff like epigenetic regulation. And for IgG glycosylation, we have a model. This is just to illustrate it. We do it in cell line. We're moving to something else, but you know, the idea is to confirm everything in vitro and in cell lines. And it actually gives good results, although the model might be a little bit crude. These are immortalized cells. It is in line with what we see in vivo. So I consider it a good model for now. And speaking of unraveling the regulatory network, you know, we had to first show, okay, we can target the executors of the glycosylation, the enzymes, but then we went up the regulatory network, and when we started changing the transcription factors, we were able to see a consistent change in glycans. So to end on a little bit of a philosophical note, glycosylation is like the third revolution in evolution. So it gives a functional analogy to coding mutations, but it doesn't have to change the coding part of a gene. It's actually just adapting, integrating information from the environment and changing the glycosylation pattern. These are the teams, GlycanAge there and Genos. That's the high throughput lab in Zagreb. And on the left is a group led by Professor Vlatka Zoldos. We do epigenetics, and I'm proud to be a part of her team. A shout to our funding sources, and thank you so much for your attention.
Thank you so much for a very interesting talk. We have a question here from...
Terrific biology. So it's like half inherited, and you said the rest is environmental. Do you have any evidence in clinical studies that any of those interventions have changed the age of GlycanAge, or there's any clinical correlate of better immunity?
Plenty.
Infection?
I skipped it for...
You skipped?
Oh, yeah, because it's like, I just listed it. But if you go to our website, there is a nice section with literature. It's a lot. We've been doing it for a decade. So menopause, that I just brushed on the highlights. The menopause, the exercise interventions, anything to do with autoimmunity, with so body weight, we see a consistent change by doing interventions. Yes, it works. We have data on it. We're collecting even more.
Unlike chronological aging, biological age is not fixed. It is often impacted by lifestyle, and it provides a much better measure of an individual's general state of health. Aside from giving you an accurate insight into your current biological age, the GlycanAge test is a powerful tool to make an informative decision about lifestyle choices and changes to improve your life quality.